Certain aquatic vertebrates routinely survive environmental extremes that would be immediately lethal to most other life forms, specifically total oxygen deprivation, known as anoxia, and complete water loss, or desiccation. The traditional view of these survival strategies is torpor or aestivation. Under this model, an organism endures environmental hostility by reducing its ATP turnover and dropping its heart rate to a fraction of its normal rhythm. The core logic relies on metabolic depression functioning as an off switch. By lowering energy consumption to near absolute zero, the animal waits out the environmental threat until favorable conditions return. Because researchers observe complete macroscopic immobility in these animals, it has historically been assumed that this external stillness reflects internal physiological inactivity. Two specific teleost models offer a unique window to test this assumption: the Anoxia-Tolerant goldfish and the aestivating African lungfish. This off-switch logic appears sound because a starved system lacks the energy reserves to maintain standard activity. However, a purely passive metabolic depression is insufficient to prevent cellular and systemic collapse. If you remove oxygen from a mammalian brain, the lack of an active defense mechanism triggers a rapid ischemic cascade, leading to massive, irreversible cell death within minutes. For an estivating lungfish, burying itself motionless in warm, moist mud creates an ideal incubation chamber for pathogens. Immobility in this environment invites massive bacterial infiltration. Effective metabolic depression requires targeted, energy-dependent cellular interventions. To prevent their own destruction, these animals execute precise defensive maneuvers at the electrophysiological and immunological levels. Dormancy is an active metabolic state deployed to prevent physical decay. In the goldfish model, the vertebrate brain has high baseline metabolic demands. It requires a constant supply of oxygen to synthesize enough ATP to function. When oxygen is removed, the goldfish is forced to rely on anaerobic glycolysis. This pathway generates ATP at a rate far below standard neurometabolic requirements. Much of this ATP powers the NAK+ ATPase pumps, which maintain the electrochemical gradients across cellular membranes. As shown in this diagram, when ATP is depleted, the NAK+ pumps fail and the neuron's membrane depolarizes. This causes the unregulated release of glutamate into the synapse. The excess glutamate locks NMDA receptors open, allowing a lethal influx of calcium ions. Without immediate intervention, anoxia causes neurons to destroy themselves through excitotoxicity. Patch clamp data from the goldfish telencephalon, collected by the Paminter lab, shows how the fish prevents this neuronal destruction. This graph illustrates the experimental baseline, using the pharmacological blocker MK801 to isolate the specific current flowing through the NMDA receptor. The secondary chart confirms that specificity by showing how high concentrations of magnesium ions further block receptor activity. With the NMDA receptor accurately isolated, researchers applied acute anoxia and charted the electrical current. At the 20-minute mark, the anoxia line drops 40 to 50% below the control, showing a rapid reduction in NMDA receptor current amplitude. Researchers also tested ethanol, a metabolic byproduct goldfish use to manage acid load, and confirmed it did not inhibit the NMDA current, ruling it out as a defense mechanism. This immediate attenuation of the receptor is known as channel arrest. The goldfish brain silences its own receptors to block calcium influx. A separate 48-hour in vivo exposure reveals a secondary, delayed defense mechanism. Once the immediate excitotoxic threat is handled, the brain downregulates its Na+K+ ATPase activity by 30 to 40% to conserve long-term ATP stores. The goldfish survives by actively re-engineering its neural permeability. The African lungfish faces a different threat: the total recession of its river habitat during the dry season. To survive, the lungfish enters an induction phase, excavating a mud chamber to begin aestivation. The lungfish executes a metabolic reconfiguration to survive for months without food or water. This chart displays transcriptomic data during induction, revealing a significant increase in gene activity in the brain. This activity produces hormonal signals, including growth hormone and prolactin, which coordinate systemic water concentration and metabolic responses. Preparing the body for extreme physical dormancy requires a massive expenditure of genetic resources. Inside its chamber, the lungfish secretes mucus that hardens into a tight cocoon, leaving only a narrow opening for its mouth to breathe air. Traditional observations often describe this cocoon as an inert physical barrier to prevent dehydration. However, complete immobility in subterranean mud makes the organism highly susceptible to bacterial attack from the soil. To counter this, specialized white blood cells called granulocytes migrate from their storage organs in the gut and kidneys through the bloodstream and into the skin. The mucus cocoon acts as an external reservoir, packed with these migrating granulocytes. Within the mucus matrix, these granulocytes produce extracellular traps to ensnare and neutralize pathogens before they reach the fish. Disabling these granulocytes results in immediate blood and skin infections, proving the cocoon is an active biological defense layer. Surviving extreme environmental stress depends on a precise reallocation of biological resources. The goldfish survives by using active channel arrest to protect its neural architecture from electrochemical degradation. The lungfish reallocates its energy to gene upregulation, entirely transforming its outer coating into an active immune barricade. These data points suggest that for these species, dormancy is a period of high metabolic specificity. These findings reveal how early vertebrates adapted to shifting, extreme climates over millions of years. Ultimately, the perceived stillness of dormancy masks a suite of complex biological responses that maintain systemic integrity against severe environmental pressure.